Hello viewers, welcome to my channel Biology Notes. Today I am going to discuss the classification and general structure and function of MHC molecules. The MHC stands for Major Histocompatibility Complex. The MHC is a large locus on vertebrate DNA containing a set of closely linked polymorphic genes. Now, these genes encode for cell surface proteins which are very important for adaptive immune system. And these cell surface proteins are known as MHC molecules. So, the MHC molecules display the antigens or fragments of an antigen to their cell surface so that appropriate T cells can recognize them and destroy this antigen finally. So, the major function of MHC molecules is antigen presentation. Three pioneer scientists, Benacerraf, Dowsett and Snell, received Nobel Prize in Medicine or Physiology in 1980 for their work on MHC molecules. Now, in human, the MHC present on chromosome 6 and it is also called as HLA or human leukocyte antigen complex. So now I will discuss the classification and general structure of MHC molecules. MHC complex produces mainly three classes of molecules: MHC class I molecules, MHC class II molecules and MHC class III molecules. Of them, MHC class I and MHC class II directly involved in the antigen presentation. Class I molecules are present on all nucleated cells in the body and also in platelets. These molecules display antigens, for example viral proteins, to killer T cells or cytotoxic T lymphocytes. The CD8 receptor of cytotoxic T lymphocyte interacts with antigens which are presented by MHC1 molecules. Then, the cytotoxic T lymphocytes trigger the cell to undergo programmed cell death by apoptosis. MHC class I helps mediate cellular immunity. Class II molecules occur only on professional antigen presenting cells like macrophages, B cells, dendritic cells, etc. The APCs are specialized in presenting antigens from extracellular spaces that have been engulfed by these cells, such as fungi and extracellular bacteria. Once expressed on the cell surface, the MHC class II molecules display the antigenic peptides to T cells having CD4 receptors. The structure and functions of MHC class III molecules are not studied well. They are not involved in antigen binding. Only few of them are actually involved in immunity, while many are signaling molecules in other cell communications. MHC class III proteins are produced by liver cells and special white blood cells or macrophages. So, our main focus will be on MHC class I and MHC class II molecules only. It is a diagram of class I MHC molecule. It is composed of two polypeptides, alpha chain and beta 2 microglobulin. This is alpha chain. It is 45 kilodalton. And this is beta 2 microglobulin molecule of about 12 kilodalton. The alpha chain is organized into three external domains, a transmembrane domain and a cytoplasmic anchor segment or cytoplasmic tail. The external domains are marked as alpha 1, alpha 2 and alpha 3. Each domain is approximately 90 amino acids long. The transmembrane domain is about 25 hydrophobic amino acids followed by a short stretch of hydrophilic or charged amino acids. The cytoplasmic anchor segment is of 30 amino acids. Now, beta 2 microglobulin has similar size, composition and organization to the alpha 3 domain, but beta 2 microglobulin does not contain a transmembrane region. It is non-covalently bound to the MHC class I alpha chain. The alpha 1 and alpha 2 domains interact to form a platform of 8 antiparallel beta strands spanned by two long alpha helical regions. The structure forms a deep groove or cleft with the long alpha helices at sides and the beta strands of the beta sheet as the bottom. This peptide binding groove is located on the top surface of the class I MHC molecule and it is large enough to bind a peptide of 8 to 10 amino acids. The alpha 3 domain and beta 2 microglobulin are organized into two beta plated sheets each formed by antiparallel beta strands of amino acids. The alpha 3 domain appears to be highly conserved among class I MHC molecules and contains a sequence that interacts strongly with the CD8 cell surface molecule found on cytotoxic T cells. All three molecules, class I alpha chain, beta 2 microglobulin and a peptide, are essential to the proper folding and expression of the MHC peptide complex on the cell surface. It is a diagram of class II MHC molecule. It contains two different polypeptide chains, a 33 kilodalton alpha chain and a 28 kilodalton beta chain. The two chains are associated by non-covalent interactions. Here, both the chains are membrane bound. Class II MHC molecules contain external domains, a transmembrane segment and a cytoplasmic anchor segment. Here, each chain contains two external domains. So, alpha 1 and alpha 2 for alpha chain and beta 1 and beta 2 for beta chain. The alpha 1 and beta 1 domains form the peptide binding groove for processed antigen. The membrane proximal alpha 2 and beta 2 domains bear sequence similarity to the immunoglobulin fold structure. Class II MHC molecules can bind 13 to 18 amino acids. The class II MHC molecule lacks the conserved residues. So this is all about the classification and general structure of MHC molecules. If you have any query, please comment. Share the video if you like. Subscribe the channel for new upcoming videos. Stay with us, keep watching. Thank you.